Hello, my name's Jodie Brunning. I'm a trustee of the Physicians and Scientists for Global Responsibility. Today, this interview with Professor Jack Heinemann, Director of the Centre for Integrated Research in Biosafety at the University of Canterbury in New Zealand, zeroes in on the work of Jack and colleagues in exploring risk nuances concerning biotechnology that is critical if we are to steward technology appropriately. Can you start by outlining your research interests and your focus, please?
Sure. Well, I'm a molecular biologist who works mainly on microorganisms, their evolution, their potential to cause disease, and most of my interest is in the development of antibiotic resistance. And that's a natural way for someone to have also then engaged with these issues of gene technology, biotechnology in general. Antibiotics were and are a kind of biotechnology. It's a way that people intervene with what occurs in nature and make an outcome more likely or reliable to happen, an intended outcome. But in their use, there are unintended outcomes. And one of those unintended outcomes is antibiotic resistant bacteria, which are then harder to treat. They're harder to treat than they were before the antibiotic because now you have one fewer antibiotic you have available to you to treat them with. So this lifelong interest, I guess, in antibiotic resistance gave me a temperament to think about biotechnologies in the light of how we use them and how we create benefit from them. And then also to take a steer from my peers and colleagues who are more in the social sciences to think about whom benefits, how those different groups benefit, and why some benefit and others don't. The issue then for me became, I guess, conflated, antibiotic resistance and gene technology, because in studying antibiotic resistance, I make genetically engineered microorganisms to understand how genes work, or to understand how the genes move between different organisms. So I'm a practitioner in the art of genetic engineering, but I'm not a practitioner in the art of entrepreneurial science. So I've never had an ambition to be commercially oriented, to seek any kind of private gain from a product of genetic engineering, other than the knowledge that I could share with the public gained from the use of gene technology. And that set me outside of many of the spheres that incentivize academics and industrial scientists who are looking specifically for a type of noun, the outcome of a genetic engineering process that can be used as a commercial entity itself. That gave me, I guess, in some circles, a desirable characteristic of being a technical expert without being technically in any way conflicted with the use of these kinds of organisms. And that has led to, I guess, 20 some years of thinking about risk and risk assessment, and also seeing how these concepts are lost within the technical community, sometimes manipulated within the technical community, either consciously or unconsciously, in order to derive certain outcomes that are preferable to them. One of them is, it occurred to me during your introduction where you said that I felt that there could be benefits from GMOs. And yes, I know there can be benefits from GMOs, but the risk of a GMO isn't restricted to the organism. What I would say is that I don't believe that the process of making a genetically engineered organism is inherently capable of only creating a safe genetically engineered organism. The process can produce either harmful or beneficial organisms. And when we talk about harm and benefit, those are contextualized. So sometimes there's harm in one place that wouldn't be seen in another, and sometimes there's benefit in one place that wouldn't be seen in another. And I think that's really quite evident in plant biotechnology. So the vast majority of genetically commercialized, genetically modified plants are in a very few number of countries at scale. And those countries have some benefit to being exporters of those products. And there are other countries that are not beneficiaries of that technology being in those countries. And there's also subpopulations within those countries that are definitely not beneficiaries of their countries producing genetically modified plants. So it can't be a single concept of benefit or of harm. And as well as that, there can't be a single concept of a product being the risk. And I think that's an important point I want to make throughout this interview, that risk is not the same thing as hazard. The product is a hazard, such as an airplane. An airplane is a hazard, but the risk of that hazard causing harm is a much more complex equation. So I would say that GMOs are not inherently safe, but there can be safe GMOs.
Can we just quickly define what biotechnology is for people that are listening?
That, is a major question. I'm writing a paper right now where the editor has asked us to talk about food systems and the role of biotechnology. And there are almost no definitions of biotechnology. Uh, but but the story gets even more interesting. So there's a few international agreements, as well as obviously dictionaries, who make an attempt to define a biotechnology. Even the Convention on Biological Diversity says that the shorthand definition is that biotechnology is a technology. Their longer definition isn't much longer, and it says that it's any kind of way that people use biological systems. So I align with that view. So a biotechnology is, for example, harvesting seed, collecting and concentrating seeds of a particular plant in a particular trait. So the genesis of corn or the genesis of wheat is a biotechnology. It's a 10,000 plus year old biotechnology, but it's a biotechnology. And in fact, it is still amongst the most important biotechnological things humanity has done in agriculture. Certainly nothing in the last 200 years comes even close to the power of the biotechnology that led to the major food staple crops. So biotechnology is a very inclusive term. It covers lots and lots of different human activities. But what we're really talking about today is gene technology, the ability to specifically intervene in the molecular structure of what we think of as a gene, and to do that at will and to our own specifications. I've looked really hard to find internationally agreed definitions of technology. Those I cannot find. I can't find them defined by patent offices, either on the national level or the international level. So everyone talks about technology and nobody says what it is. We think, or I think, technology is that means to do something that otherwise we would have to wait to happen spontaneously. Archimedes said that if you gave him a big enough stick and something to balance it on, he could shift the earth. So that was his lever. He could shift the earth. The lever is a technology. And otherwise, he would have to wait for the earth to shift when and if it ever did. So a technology is what gives us the ability to do something at pace or at will or at a particular size. And as such, that technology has inherent within it the ability to produce something at will that we want. And it has all of those things we can't control that produce by our will things we don't want. And every technology has that. A small number of technologies become safer the more you use them, but the vast majority of technologies create harm at some factor, some sub-factor at which they create what we perceive as the benefit. An example of a technology that creates safety the more it's used is a seatbelt. So if I wear a seatbelt, I reduce the chances of being harmed after I've lost control of my automobile. But in addition, I decrease the chances of losing control of my automobile because I'm constrained in my seat and I can still operate the steering wheel and the pedals. So conditions that would have caused me to lose control in the past no longer cause me to lose control because I'm wearing a seatbelt. So that increases my safety. But also, if other drivers are wearing seatbelts, it also extends the range of conditions under which they can control their automobiles. So the more of us who are wearing seatbelts, the safer the road is, the safer the operation of my automobile is. That's a rare example of a technology whose increased use results in increased safety. The vast majority of technologies increase output, but also increase the potential for harm. And that's why we have risk assessment. And that risk assessment takes into account the fact that something is a hazard. An automobile is a hazard. A GMO is a hazard. But whether it causes harm is the complex equation of its potential to cause harm times it being in the place where it can cause harm. And that place isn't just its location. Its place is, does it have enough of it to cause a harm? Is it being in the range of those who are susceptible to harm? So a type of hazard that might be very important for children, but unimportant for adults, is the risk of that is low if it's never found in a place with children. And its risk is high if it is found in a place with children. So its risk is not the same thing as it being a hazard. I've forgotten why we're on this, but that is that is the nature of a technology, and a biotechnology is just those technologies that involve life.
You quote Jansen et al. Biotechnology is the skills, tools, knowledge, and techniques used to accomplish certain ends. And so you're starting to, you're drawing attention to scale and the actual technology itself. And part of this discussion involves looking at precision, the new gene editing tools that are changing how biotech developers, use biotechnology. How does that, the precision claims or the precision statements, how does that sort of weave around your discussion here?
Precision, I think, is a manufactured word for the community, the biotechnology community. It's long been used to try to placate concerns that the unexpected will emerge from the process of making a genetically engineered organism. The unexpected can emerge. No matter how sophisticated our techniques become, there are still things we can't anticipate. And in fact, we're learning what we can't anticipate as we work on the development of the techniques themselves. But this shouldn't come as a surprise, but it is a term that for which there are other words that would be less emotive for the purposes of manipulating the public that they could have chosen to use if the intention were not to manipulate. What each of the techniques allows is for the intended outcome to be achieved easier, which is a scale issue. If it can be done easier, you can make more of them, or you can make the same number faster. So it is an efficiency. Those types of techniques make it possible to achieve the intended outcome with greater efficiency. And that's entirely an industrial concept. It's a monetary concept because it constrains the dimensions of cost, time, materials, whatever it might be. The techniques increasingly become independent of a laboratory because inefficient techniques give you what you want, but it's harder and fewer of the times you do it. Do you achieve what you want? You need a laboratory to keep out all of the contamination that would obscure that result. We think of laboratories as keeping the world safe. Laboratories weren't invented to keep the world safe. Laboratories were invented to make our technologies occur with greater reliability, the outcome occur with greater reliability. And a secondary property of them is that they contain anything we make that might be harmful. The more efficient our techniques become, the less dependent we are on the infrastructure of a laboratory to get what we intended to get. That efficiency is not the same thing as what most people would think of as precision. So precision is substituted for the concept of efficiency to say that we also decrease the number of things we didn't want to get. And there's no evidence for that. So we always get unintended side reactions in any biochemistry in a gene technology technique. So the more often we use that technique, the more unintended effects we create.
Can you provide an example?
If I were to genetically engineer a plant to be resistant to a herbicide, and I do that in a laboratory and I use a highly efficient technique, I can isolate a bunch of different possible plants that are my intended outcome. And maybe all of them are my intended outcome, or maybe 1 in 100. That's an efficiency claim. No matter how efficiently I get that, it doesn't tell me how often that technique caused a change somewhere else in the genome. It may have also done that with much greater efficiency because of the nature of the biochemical reaction than if I had not used the technology or done a different technique. I might get that one less frequently. So maybe I get that only 1 in 150 times. However, I don't know which isolate will and won't have that unintended change, if indeed none of them ever would. Because, well, a biochemical reaction occurs over a space of time, and the chances are that there are changes I can't anticipate, and unless I look really hard, may not find. Every time I repeat that process on the product of a previous process of using my techniques, I increase the chances of yet another unintended effect. And that's precisely what the new techniques allow. The new techniques allow me to make serial changes in the same organism over time, and thus accumulate a legacy of unknown changes. Now, I'm not saying that certainly not all, or even any of the changes may be harmful. But by thinking they're not harmful doesn't make them not harmful. And that is the whole purpose of regulation. Regulation works at scale transitions. So where harm can accumulate at scale transitions, that's precisely where regulation is a solution to mitigate risk. If we go from the laboratory to outside, that's a scale change. In the laboratory, I had a sterile environment where I knew that only my plant was exposed to my tools. If I now do that same process, which the new techniques allow me to, if I do that same process outside of a laboratory, say in the field itself, there are billions of organisms exposed simultaneously. All the microorganisms in the soil, all the microorganisms on the plants and in the air. There are animals, invertebrates, mammals that are running through these fields, birds. All of those things are simultaneously exposed and have the potential to react to that same biochemistry. And every one of those changes is unintended because they're not even my target organism. So at that scale change, regulation would really matter. If we were to say the technique itself is inherently safe, and therefore no product has to be regulated, then I can use those techniques outside in the field. By deregulating the hazard, which is the organism I intend to change, unless you have commensurate regulations on the process, you deregulate when and how people use these techniques. They can use them anywhere. They can use them in schoolyards. They can use them in the center of the city. They can use them in their garage. So we have to think in that trajectory of the technology, not in the end point of the technology, to understand why regulation is an important assurance for society that the biotechnology product, not just the technology product is safe, but that it didn't create a hazard in its making.
You're really good at explaining and simplifying sort of what a lot of us intuitively know, but we're not able to explain it. Being as simple as saying that biotechnology speeds up biological change in comparison to natural evolution, and it amplifies risk or the potential for risk. Jack, you're the director of the Centre for Integrated Research in Biosafety at the University of Canterbury, New Zealand, and your latest paper is the result of a collaboration between University of Canterbury researchers and its colleagues from the Climate and Environment Division, NORS, I think, the Norwegian Research Centre. And now I consider this latest paper should firstly be essential reading for scientists, scholars and students who research and work in biotechnology. And this work adds to a compendium of work on scalability and risk that deepens the understanding of how we can legally, socially and culturally understand much of the tech. And this sort of can, for those scholars, can also, I believe, encompass the digital technologies because, of course, going back to our discussion on tech. So this can then inform ethicists, sociologists, philosophers, and even economists. Now, because the Null segregates paper discusses a risk that is not often regarded as a risk, it's an excellent case study to assist students, teachers, and educational institutions to recognise the nuances that can easily be dismissed by powerful operators and perhaps regulators that are more informed by their sponsors or their the industry applicants than by independent scientists. Can you please explain what a null segregant is and briefly outline this paper?
Sure. A null segregant, and in some places they're called negative segregants, is something to which DNA has been added, and that added DNA has been removed. So they're the Schrodinger's cat of biotechnology. They're both a GMO and not a GMO at the very same time. And that's a conundrum. So if you believe that genetic engineering can only be a process in which DNA is added to an organism, if you believe that, and I don't subscribe to the idea that that's what a GMO is. But if you were to believe that, well, then that organism becomes rehabilitated, remade, reborn as something that isn't a GMO by the loss of the DNA. In the 1970s, when the first regulations of gene technology were made, and those first regulations appeared to us to have emerged out of the UK in about 1978. The way in which these techniques were used became regulated, and that was now referred to as process-based regulation. The US initially subscribed to that, but diverged in the 80s to what is often now called a product-based. To put in terms of our earlier discussion, the US attempts to regulate hazard, and Europe, which adopted the UK vision, and then the Cartagena Protocol, which is the international agreement on regulation, went for a process type description. Even that's oversimplified. The US also uses process to inform what products it regulates. And in the European Union and Cartagena-based countries, the product is assessed. So there is product and process in everyone's regulations. But where it matters is in definitions. What becomes regulated is determined by whether or not it is defined as coming through a process or it's defined as a hazard. And the US uses a hazard base. And that hazard based viewpoint has become almost, I wouldn't say almost, has become infectious among genetic scientists who very much align with the idea that if you can make something in the laboratory that could appear spontaneously somewhere on Earth at some time in the last 4 billion years, then there is no reason that it should be separated out for risk assessment. And that is conflating risk with hazard. In the same way that I would say, a uranium atom found in the deserts of Australia will emit radiation spontaneously, and that's natural. And the same decay pathway may occur at the heart of a nuclear bomb. Doesn't mean I deregulate nuclear bombs. If I just look at the hazard at the atomic level is identical. But certainly the scale at which that hazard exists at the heart of a nuclear bomb that's been detonated is very different than the scale in the Australian desert. And where it detonates will be very different exposure pathways than it is in Australia, because it's unlikely that a nuclear bomb will be detonated in the deserts of Australia. If it's detonated, they'll be at the heart of a city where people are. So this whole conflation of naturalness, the idea that something that can occur without human intervention should not be regulated if it does occur by human intervention, is artificial. And it is a fixation on just a small aspect of the risk assessment, the hazard level, rather than risk, which is a combination of the inherent potential to cause harm times it being in the right place and time to cause harm to something that you're trying to not cause harm to. And that's what risk is. So many products of many GMO products in the future are going to be null segregants. And the reason is that the techniques called genome editing and similar techniques involve putting in a piece of DNA, causing the rate of genetic variation to change, so mutagenesis, the pace of mutagenesis to change. And because they are biased sources of mutagenesis, they occur with high efficiency at the intended location. That doesn't mean they don't occur with low efficiency at unintended locations, but they definitely occur with high efficiency at the intended location. So the rate of change, genetic change, is much higher during the time that this new piece of DNA is in the cell of an organism. Once you get the change you want, you lose that extra piece of DNA, but preserve the effects of having caused mutations, amplify the organism to a commercial scale and release it, or you'll do this right in the environment where it's already released because the techniques are that efficient now. All of those things are called null segregants once they lose the piece of DNA. And the idea is that, well, mutations can occur in nature anyway, and we don't regulate the mutations that occur in nature, so we shouldn't regulate those that we create with our technology. But once again, the point is that when we do it at a scale that doesn't occur in nature, or almost never occurs in nature. And then on top of that, we do it in a place and to organisms that we are exposed to, or particular environments we want to protect or species we want to protect in those environments. We do it in those places that the same effect in nature would occur extremely rarely, if at all. So we change the scale of exposure, which is the other part of the equation of risk. Null segregants are a big part of that biotechnology imaginary, and that is how we come to understand how a biotechnology works.
So can you just quickly talk about biotechnology imaginary, because it sounds a little maybe fluffy to people?
Right. Yeah. So and pejorative, but it's not. Well, sometimes it is when it comes from me, but it's not intended to be pejorative. It is how a group understands something. So as a technical expert, I have a biotechnological imaginary about how something works. And other groups may have the same or a very different imaginary about how something works. When it comes to risk, then, it becomes really important how we think something works. If we think something works precisely the same in my laboratory as it does in nature, I'll have a different concept of harm, potential for harm, than if I think they work exactly the same way. So the difference being that if I do it in my laboratory, there's very different exposures. So even if there's identity on the molecular level, there won't be identity on the evolutionary or exposure level. And this is an important point. Evolution has two parts to it. So in nature, there's all sorts of ways in which genes change. And the biochemistry of that change may be identical to how I cause change in my laboratory. It could be. And even where it's different, that may not be different in a meaningful way. So that is the source of genetic diversity. But the far more important part of evolution is selection. So even if you get variants, what happens in nature is that those variants are fit, they're more fit or less fit than somebody else next to them. And if they're fit enough, they remove those who are less fit. And that's called evolution. It's a change in the frequency at which a trait occurs in a species. We become the agents of natural selection when it comes to gene technology. While we might hypothetically have identical inputs as nature into the generation of diversity at the molecular level, what is completely separated from nature is how we select for the outcome. And that is where risk matters, because by selecting, I change the scale. That is the evolution of that individual, because it becomes a population and the frequency of the traits I created change. So that's the important point. And many, I think, of my molecular biology colleagues have forgotten that evolution isn't mutation. It isn't just creating changes in genes. It is that really important process through which selection changes the frequency of a trait in a population. And we are that agent. And that is what's not captured if you only look at hazard.
Yeah, and my undergraduate degree is agribusiness, and so I'm very interested in and soil. So if you imagine that you've slightly and subtly changed unintended effects that have changed these traits, you could easily plant a crop that where it previously perhaps zinc uptake was fine or nutrient uptake was fine, that's then sort of slightly distorted, so the crop isn't as fit as it might have been in the past. But because this is a chronic kind of issue, people can't really identify that. Does that make sense to you?
Yeah. I mean, look at the southern corn blight epidemic in the early 1970s in the United States. So early in that decade, the world was on the cusp of a famine because of a projected loss of nearly all of the US corn crop that year. I think it was 1972 or 1973. The reason is 50 years prior to this, the invention of a very efficient way of generating corn hybrids was made. And that led to the biological patent on maize. So we learned how to make hybrid varieties by crossing two different elite lines. The offspring do not recreate the desired traits. So those seed producers could hold the property exclusively because they owned the elite varieties. And the entire corn seed industry changed to producing hybrid corn for sale to farmers. Shortly after that invention, a natural mutation was found in maize that caused male sterility. The line that gives you male sterility could be substituted for one of the elite lines, and you reduce the labor costs of creating your seed stock. So one variety became inbred in nearly 100% of the US corn supply. The output was a massive increase in yield over many decades. But what you couldn't see is that 50 years into the future, there would be a pathogen who was specific to that variety of corn. And the conditions for that pathogen emerged in the early 1970s when there was no substitute for that variety in the industrialized process of creating hybrid corn.
And
So the trait we selected, that we substituted for the wisdom of evolution to make, evolved a line of corn and a food system that was entirely dependent on this monoculture of one elite parent and almost resulted in a worldwide famine. Now, what saved us was a lucky change in the weather. That gave breeders enough time to come up with another variety that was male sterile to maintain that industrialized process. So we learned nothing about our dependence on a highly efficient industrialized process for creating our food lines, but we were saved from famine that one time.
thank you for that explanation. I'm going to just to reinforce what you've been saying with a quote from your latest paper. You said and colleagues said, the generic value of gene technology is its scale gearing to deliver the benefits it promises at commercial time and production scales. The source of inseparable potential for harm is this same property that creates hazards at scale. The gearing increases with newer techniques. So I like the concept of scale gearing. It does flesh out this work you've been doing.
And that is a concept of efficiency. So the scale gearing is greater efficiency.
And you've also pointed out that deregulation of the intended null segregants may de facto deregulate the organisms, e.g. Bacteria, fungi, and invertebrates exposed to the technology along the way of developing the intended null segregants. Can you flesh that out a little bit, please?
Yes. So if I use these techniques in a laboratory, and I was saying this earlier, my laboratory is sterile. Only the intended organism is exposed to my techniques. And then even among the many replicates of that organism that I look for candidates that I want to keep, everyone else is disposed of. Everyone else is killed. So only what I intended to make or intended to save from the process is retained. The techniques have gotten to the point, though, where their efficiency is so high, I don't need to do them in a laboratory. Well, in fact, during the pandemic, researchers in the US were both developing and then sending out to school children the kits to do genome editing in their kitchens. So what that meant was you had people who were not trained in laboratory work to protect themselves, others, or to maintain a sterile environment, doing it where people eat, and exposing all sorts of microorganisms, including the potential fecal coliforms on their hands, the streptococci that inhabit their noses, the yeast that are growing under their fingernails. All of these organisms were being exposed to these reagents at the very same time as the organism intended to be exposed, which was part of the kit. And to a range of people, from young children to their parents, who may have been in that same lockdown household. Now, that's just an example of how these techniques have become of the efficiency that you can do it in a kitchen and you can see an intended result, but you still are not able to eliminate unintended results, not only in the target organism, but in all the other things that are exposed at the same time, that you don't any longer control. So that is exactly the point of an efficiency increase. And I want to make something clear to my colleagues who sometimes misunderstand this pace argument. They confuse the idea that these techniques have become more powerful because of their efficiency with them delivering a benefit to society. That is not the same thing. So they increase the efficiency at which I get the change I intended to make. But still, how often one of those organisms is a benefit to society comes down to how smart I am. And for the most part, we guess very badly. And when I say we, I mean the science and technology community. We rarely make a GMO that actually makes a difference when it's commercialized. And we never make a GMO that doesn't have some trade-off once it's commercialized. So the techniques don't bring benefits faster. The techniques bring the intended changes faster, and we are still as inefficient at knowing what we need to change as we ever were.
Yeah, and I'm seeing that, I've had children go through high school recently, and the positivistic, the positioning of biotechnology.
It is definitely a double standard. So if you are rabidly pro, and often meaning that you are, aligned with commercial interests as well, or those with commercial interests, the criticism is smaller than if you're rabidly anti. And also the punching down is decidedly different. But that's been true for technology and the conflict of technology in society for a long time. There have been lots of books written about it. In the 90s, there was a great book called Trust Us, We're Experts, looking at such things as, mining effects and other conflicts of science and technology with society. The arrogance of it is then reflected in that polarization, but only one side of the polar divide is blamed for it. And I also find that frustrating. I find it, it's of course challenging to interact with people who disagree with you. And if they don't share your language and experience, it's even the more frustrating because it's hard to know when you're talking about the same thing or what you have agreed to as a common assumption base. But the inherent idea that it will be, or I should say, the paper you mentioned, we wrote that paper on how biotechnology is seen in societies and for climate change. And that was part of a project that we had done with the IPCC and was in the last IPCC report. And the the issue there was once we wrote that paper and we said sometimes just the production factory of promises from biotechnologists is harmful because it distracts society towards delaying action while that promise is supposed to be fulfilled. More often than not, it's never fulfilled. leaving us with the same problem, but also a loss of opportunity to have solved it, and the exacerbation of that problem over that time. Whereas if you had a very different way of structuring your technology and society, you would come to different solutions. Um, in reaction to that paper, someone said, "Well, I'm pro-technology." and and um, it's perfect because the paper didn't say technology was antithetical to solutions. It said that the technology promise mill was often conflicting with the benefits that it could provide. And really, the snake oil, we call them snake oil salesmen for a reason. The same thing happened in that period of time in the early part of the 20th century when everyone was promising the famous elixir that would cure all ills. It's the same way we invest in biotechnology, and whether it's a government or it's an angel investor.
And I can understand how this is great. Scientists love the work, industry love the income, politicians can tell the public that they're onto it. So we have this technology push. They're doing something. But if you don't have the principles, the values at the head that can say, this is how we want that technology to work, this is the broad problem. What we do is we basically incentivize the people that have the greatest financial capacity to then say, we're going to solve that problem for you, without drawing attention to all the myriad of sort of social, cultural, other economic issues that may have been building up for years that are then left out in the cold. Drawing attention to that habit, the pressure from developers, you've called that technology push versus goal pull. Do you want to just outline that a tiny bit more?
Yeah. So what we do is we distinguish how something would work in two different scenarios. Technology pushes the status quo. We let technology providers also tell us what our problems are. So often, a chemical giant, chemical corporation will tell us that our problems are weeds, and therefore we need a solution, which is a herbicide. And then it will tell us that the problem is herbicide-resistant weeds, and therefore we need another herbicide. And we will need a GMO plant that we can spray with the herbicide. So they define for us what the problem is. We may agree, or certain groups may agree. And then they do, because they also have in mind what a solution is, and because they provide solutions of a certain type, technological ones in which intellectual property can be controlled, uh, those problems are defined in a way that suits their hypothetical and sometimes actual solutions. Governments fall prey to this. They will define a problem in such a way as it allows for somebody to invent a technological solution, and even if that's a hypothetical one, and then governments invest in a research and development grants, or others, private sector invests. In any way, what we've done with technology push is we've monetized promises. Promises can be sold in the same way that widgets can be sold. And in fact, there's probably a really high profit margin for promises and a much lower one for widgets, because the vast majority of promises are never realized. But we forget when those promises have gone unfulfilled, and the same purveyors of the promises have moved on to other promises, either for the same failed technology or for new ones that will fail in the future. So we don't have any feedback loop that hones, evolves a technological approach that's rational. The contrast to that is goal pull. Goal pull, though, is highly susceptible to being misused. And when it is, it results in the same thing as technology push. So the key thing for goal pull is for a society or a decision maker, somebody, to identify an authentic problem. To contrast the two, we saw this. Let's say what you think is that food is going to be scarce. Technology push then leads to all sorts of promises like plants with enhanced photosynthesis. So these plants can be genetically engineered to more efficiently turn the sun's energy into biomass. So that's a technology push on the problem of food scarcity. If you are a goal pull and you adopt the same problem, you may come to the same fallacy, and that is you need more production of plants. Food scarcity, let's grow more food. But the problem isn't food scarcity. It isn't a problem ultimately that can be solved or necessarily solved by productivity changes. The problem is that not everyone gets enough good food. So it's not food scarcity. It is that everyone is not getting an adequate amount of the right food. When you define the problem that way, then you can ask the question is, if everyone got the right amount of food, do we produce enough for that to happen? Now, the answer turns out to be yes. In fact, we produce enough good food on Earth right now to feed 12 billion people without a problem. So the problem isn't one that requires a technology that changes production. The problem is at its root a social problem, social justice problem, an economic problem, and in some cases may be in a local situation a productivity problem. But once it's defined, once you defined the authentic problem, you find that your solutions tend to be a broader palette than just one in technological invention or another. That doesn't make it easier. We haven't been able to solve the social problem for a long, long time. But the promises of technologies don't incentivize us to try. So if governments think that there can be a dramatic change in productivity because of a genetically engineered plant that can harvest more solar energy magically without all the other inputs that plants need to grow greater amounts of biomass, if they think that, then they may not undertake the difficult work necessary to change the social conditions that lead to a maldistribution of good food to everyone on Earth.
I'm rather, interested in the concept of safe and nutritious food. So, what is safe and nutritious food? Does that fit in the informational networks that sit inside our governments that lead to discussions around what technology is appropriate? And I don't believe that happens within our governments or within, say, societies such as the Royal Society. And what you also tend to get is you get elites, and of course elites is a scientific term, that because they have such knowledge in a particular narrow area, they then control the paradigm of what moves forward. And that's, of course, a very Kuhnian problem. You end up with a sort of a dogma of what must get done and what can done. And this seems to be an eternal problem where, science and the governing of science is sort of distorted or corrupted. Does that happen? Do you see that happening broadly, or is it
No,
just?
yeah, of course it is. And that's what the elite is what I call the technical expert. So it's emphasis on technical. I mean, in some ways, it's been my entry card, at least initially, to some fora because I'm a technical expert. I'm a practitioner. And I wouldn't have even gotten that far if I hadn't been a practitioner. They may not like what I have to say afterwards, and I don't get invited again. But I get in the door because I'm a technical expert. Also, when I have challenged the EPA, I recognize, as unsuccessful as I am in challenging the EPA, I get much further because it's harder to dismiss my knowledge of the science than it would be for other publics in New Zealand society. And the problem, though, that even though it gets me in the door, is that I encounter other technical experts who are not necessarily more expert in the tech, but don't need and have never needed to engage in the social problems in which their tech's embedded and don't necessarily have the skills or the temperament to think about solutions that wouldn't also addict society to an ongoing need for that technical expert. So the inherent conflict of interest is one in which we secure our position in society.
The problems, the social problems we see are very local, where we have hunger in New Zealand, yet 80% of our food is exported. And this speaks to the Irish potato famine, where food was continuing, the most nourishing food was being exported. It was just the generic potatoes that they could afford. So the local factors shape what we value and what our norms are. And so this should be really important to how we steward and judge what is risk and how we steward health. It's going to haunt us until we actually turn around and start making a place for science that is independent of that need to produce IP, to produce what we call innovation, and that's my master's research project looked at that difficulty.
Yep, I would put just one more thread in that innovation bow. One could even accept, if one hypothetically accepted that innovation itself was a good thing. Okay, it comes down to then what we decide to define as something that can be protected by an intellectual property right, rather than innovation per se that determines what's available to us. So as I was saying earlier, the most important innovations in human evolutionary history, things like the domestication of wheat and maize, etc., none of those could be protected by modern intellectual property rights instruments. And nothing that has been protected by those instruments is anywhere as near as important as those things to what we have and what we are. So it really comes down to the heart of the matter being that what we define as our priorities determines what we will get. And if we are left to define our priorities, we almost certainly will define them in a trivial term. So we get things like cars and cell phones. I mean, that's also a classic example of a technology that we didn't know we needed, right? So in the 80s and 90s, we weren't going around thinking, "Man, I wish I had Facebook." And then once we had it, it became addictive. The issue I guess I'm trying to get at is we don't necessarily plan out the most important things to reward. And unless we get, and that's what I mean by the authentic problem. We have to have authentic priorities, authentic problems, not ones that are conveniently just an invention away from satisfying us.
Jack, honestly, thank you very much for your time, Professor Jack Heinemann, University of Canterbury. I do emphasise that people should go and look at that Null segregates paper. I'll provide a link to it at the end of it. But thank you very much. It's been a pleasure talking with you.
It's a pleasure. Thanks for having me.
Thank you.